DNASTAR protein tools provide a wide range of analysis and modeling options, and Thomas Leary is going to show you a couple examples of those. And what I'm going to do here is just give you a kind of a high-level overview of what some of these tools are capable of, so that if you want more information or want to do a webinar with your colleagues more specific to your research and your needs, we can set that up. So I'll just give a kind of a very quick overview, and then the majority of the webinar will be Thomas showing us a software demonstration, as Sharon mentioned, with the novel coronavirus spike protein structure, and using that to illustrate some of the capabilities of the software, including structural analysis and biophysical properties. I know we had some questions come in about that, looking at epitope identification, some visualization and rendering to make your structure look the way you want it and even get it ready for publication. We also have tools for protein alignment, as well as protein design and modeling. And I think we'll also be briefly showing some protein structure prediction with our NovaFold application. There's a lot to cover there. So again, if you want a little more in depth on protein modeling, I would say to schedule a follow-up webinar with Thomas after this. And then at the end, we'll do as much Q&A as we have time for. So Lasergene Protein, if you're not familiar with it, it's really based on our Protein 3D application, which has some really powerful visualization tools, as well as sequence and secondary structure analysis. And all of those capabilities are integrated. So for example, you can apply epitope prediction methods and then visualize those epitopes on a rendered surface on your protein. We also have some protein design capabilities. Thomas will show some of that today. And then protein modeling, which is a big field in itself: protein structure prediction, antibody modeling, and protein-protein interaction modeling. If you're familiar with our genomics or molecular biology tools, there is some integration between our protein tools and those. So for example, if you're doing NGS analysis and you have variant data, our SNP to structure workflow actually allows you to model those variants on the protein structure. So you can do a lot of filtering in Lasergene genomics to find variants of interest, whether they're disease causing or predicted to be damaging. And then also take that information and model it onto the protein to see, could it also be destabilizing to the protein structure itself. Another place where we have integration is protein design and site-directed mutagenesis. So if you're familiar with SeqBuilder Pro, perhaps you use that to design PCR primers. You can actually mutate those primers in SeqBuilder Pro and again, see the impact of those mutations on the protein structure before you do your experiment. So I mentioned protein modeling. We have three what we call NOVA applications that do protein modeling. So the first is NovaFold, which does protein structure prediction. The second is NovaFold Antibody, which also does structure prediction, but specific to antibody structures. And then NovaDoc models protein-protein interactions. And so one example of how you could use all three of these applications together might be modeling an antigen with NovaFold, an antibody with NovaFold Antibody, and then modeling the interface between those two with NovaDoc. A little bit of background on NovaFold. This is based on the iTaser algorithm out of the University of Michigan. We are the sole commercial distributor of NovaFold, meaning if pharmaceutical companies or commercial labs want access to iTaser, you would get NovaFold. We have additionally made some improvements to the iTaser algorithm, and we also do some customization. So if you need some customized work done on the templates or on the results, we can help you with that. So iTaser and NovaFold are unique in that they use a hybrid threading and ab initio modeling technique. So it uses template structures as well as a de novo approach to model regions of the structure which can't be resolved with the known templates. And I think Thomas will show a little bit of how that can be applied in his demonstration. As I mentioned, we can do some custom work with NovaFold. So we have a nice case study on our website where we modeled a difficult to model GPCR structure using a combination of standard template libraries and custom libraries, and then aligning those structures to get sort of a hybrid model that has the best of both worlds and better resolution. And I think I have the URL down here. It's on our workflow page. I think Sharon's going to show you how to access those pages at the end of the webinar. But if anyone's interested, we can also send you this case study directly. NovaFold antibody is strictly a homology modeler, and so that uses a library of 1,800 antibody models for templates, and it's super fast. The predictions can be done in, usually just a few minutes. And then NovaDoc, as I mentioned, is used to simulate protein-protein interactions, and it's based on SwarmDoc from the Francis Crick Institute. DNASTAR is the sole licensee of SwarmDoc, so if you are interested in trying that, NovaDoc is your best bet. It'll not only give you access to the SwarmDoc algorithm, but also really nice interface and reporting. And SwarmDoc is unique in that it incorporates motion and allows for backbone flexibility during docking. So again, some unique attributes of the algorithm really designed to give you the most accurate models. So that's just a very quick overview. I know we covered a lot. At this point, I'm going to hand it over to Thomas, who's going to show you some of these tools in action using a SARS-CoV-2 spike protein as an example.
Okay, I've just handed that over to you, Thomas. I see that you were purple for a few seconds, but your webcam looks like it's showing the correct non-purple skin tones. Thomas is not an alien from another planet. So anyway, you are ready to go now, Thomas.
Perfect. Thank you, guys. Thank everybody for coming and joining us for this webinar. We really appreciate the high turnout. They're very excited about the last few weeks. I think that we've all been kind of stuck at home. Unfortunately, I've been one of those few people who get to come into the office. Some of the work that we do was determined to be critical in Wisconsin, and so we were allowed to stay open and functioning for the past few months. And today I'm going to show you a little bit about some of the things we've been working on, and then also at the same time we're going to look at this. I want to introduce to you the 6VSB structure, which is the first cryo-EM structure to show up in early or late February on the PDB that represented the spike protein, the one of the targets that we're all dearly looking into, trying to figure out how we can develop vaccines or antibodies against this virus. But anyway, let's get started. So right here you can see my screen. I have I had the 6VSB spike protein loaded up into the interface. And the way I got it loaded up, just so we start from the beginning, is I went to the file menu and I went to open from PDB. So if I go to open from PDB, I can type in the PDB ID of any protein that's in the PDB, and it'll automatically download it for us, which is a nice feature. It lets you quickly get access to different proteins that you may be interested in. And so that's one of the ways to get files in. But secondarily, you can always just drag files in. So for instance, here's a newer PDB of the RBD, the receptor binding domain, bound to the ACE2 receptor. That was released about a month ago into the PDB archives. But you could just drag this in like that right into the menu. It'll automatically open it that way. So that's one way that I like to open different files, is just dragging and dropping. You can always go to the file menu and open, but generally I don't do that. But I'm going to jump back to this 6VSB model, and I want to show you a couple things about the NovaFold or the Protein 3D interface that can help with some of the work that you're doing. Now, also, a lot of you may be working on NGS projects or Sanger sequencing projects, looking at variants in the spike protein and finding, getting ABI files or NGS data. Now, one thing to consider when I show you all this information in this workflow is that all of that data can be used to further refine models that we have. If we can create a predicted model and then you can find some variance in your Sanger or NGS data, we have a modeling workflow here that lets us model those variants into the structures. So that's one way we want you to remind everybody that the interconnectedness of our molecular biology experiments and also the protein and structural biology experiments is that we can really help transition you to being able to view the rotamers of those variants that those non-synonymous variants that you're detecting with the SeqMan Pro or SeqMan Ultra tools, or with our SeqMan and Gen product, or any other ways that you're doing your sequencing. So just bear that in mind, that we're going to look at this protein design component, and the information we will use here will be coming from your NGS data or your Sanger data that you're producing in the lab. But anyway, so one of the first things I want to show you is if we go over to the right here, we can see there's an Explorer tab. So the way this tool is set up is that it's got a series of different tabs and panes that we want to use for a number of different things. So we have an Explorer tab, a Features tab, that's where we show features. We also have a Tracks tab. The Tracks tab lets us control this biophysical properties graphs here on the left side. And this is going to help us see things like Jameson-Wolf antigenicity, Kite-Doolittle hydropathy. We also have charge density, flexibility, B-cell epitope predictions, T-cell epitope predictions, MHC2 epitopes, as well as something I'm going to show you quite a bit today is how we can use cleavage sites to investigate this novel protein, because there's some interesting cleavage sites that are introduced by by some mutations. I want to be able to show how we can use those cleavage site predictions in this workflow today. Additionally, I'm going to be using this coloring tab here under the style menu. And so this is also part of the pane interface. We have a details pane, which we'll use to show some RMSD information. And then here's the style tab, and we'll use that to change colors of our surfaces and create different regions so we can view this structure better. So I'm going to jump into it. So what I'd like to do first is I'd like to show you how we can apply a molecular surface initially to this structure so we can get a little bit of a better view out of it. And so what I've done is I've selected the A, B, and C chains here just by shift clicking or a control click. And then I'm going to go down to this surfaces tab, and actually it's normally closed like this. You'll have to open it like that. And then if I click here, we have a number of different options. We have molecular and we have solvent accessible surfaces. I'm going to show you how we can do a molecular surface initially, because then it'll give us a little more granular view of the surface topology of this protein. And so I just applied a surface to the entire trimer, the A, B, and C components of the spike protein here. And then also I'm going to go down to the fill color here, and I'm going to change it all to gray because I actually want to have it a nice neutral color. I'm going to start painting this for you to show you different regions where epitopes are, where there's regions of hydropathy, and where there could be MHC2 complexes on this protein. Additionally, if we look closely here, there's a number of glycans that are coming through on this model. So this is part of the cryo-EM structure model. You can see here that there's 62 N-acetyl-D-glucosamine glycans bound. Now these glycans, it is my understanding, they play a big role in many parts of protein's life. First of all, they help with solubility. Also, they may be protecting certain sites that are highly antigenic. So let's remember that these glycans are extremely important, and this is one way for us to understand how to use them. Now, also, I'm going to show you how we can also, when we have a predicted model, we can use these glycans to further refine our predicted models, because we can show them overlaid on top of the template models. They'll have those glycans present. And so one of the things I want to do next is just quickly color the trimer so you can see the A, B, and C chains more clearly. So what I've done is selected the A chain here, and then I'm going to go down to the fill color, and I'm going to just change that to orange. And then I'm going to go to the B chain, and I'm going to change that one to, we'll go to purple here. And excuse my color choices, sometimes I am colorblind. And then I'm going to go to the C chain, and I'm going to apply a third color here, and we'll choose maybe this. That'll look good, magenta color. And so this is the trimer, and now we can see it in better detail, and we can see its symmetry, which is interesting. Now, also, we are looking down on it, actually. So if you imagine, this is sticking out of the top of the nucleocapsid, I believe. So this is sticking up. And so it looks kind of like an upside down pyramid if we look at it that way. And also, one thing to note is that the A chain, if we go over here to the A chain has one of the RBDs, which is the receptor binding domains. It has it in the up position. And so that was described in this model, in this 6VSB models publication. But the RBD in the up position is an interesting flexible region, because it is my understanding that 2 RBDs have to be up for it to bind to the ACE2 receptor. For it to bind, after it binds to the ACE2 receptor, is when the cleavage can occur to expose the fusion protein. Now, this is the way I understand it. Obviously, everybody may have more information on it, but I just wanted to point that out, that this model does represent it with the RBD in the up position. Now, this model was released 2/26, 2020. Now, that was about 26 days since I initially heard really about the situation. I was in an airport, and there was warnings going on in the airport about exposure. And so that was 26 days after that event when this model came out. Now, this model has a couple issues with it that I want to point out, that's interesting, and how NovaFold, the tool that produces our structure predictions that Katie was mentioning, can help assist with the issues that this model had, and how we could have had a better chance in the beginning by using a workflow as such that I'm going to present today. So if we see right here, this is the RBD, one of the RBDs, but if you look here, there's these fragmented edges. Now, those fragmented edges right here, I believe, are from the cryo-EM was not able to pick up the atoms in those regions. And if we go to the sequence view, you can see that in that region, there's these grayed out areas. These grayed out areas are simply regions where the cryo-EM did not find any atoms or wasn't able to resolve them. They were too flexible. And so we're missing those regions in this model. So this was on 2/26/2020. But this was we were excited to see this model because we didn't have any idea of the exact topology of the RBD, and we were really interested in that for some projects we're working on. And so we used our NovaFold tool to remodel the monomer, so just this A chain of this spike protein, and we did that. We actually did that about 6 days before the release of that model we were just looking at. So here's my run. This is the Nova Cloud Services pane. This is where, if you submit a prediction via our protein structure prediction interface here, this is where the results show up. It gives you information on the date, the time of the run, the model accuracy, the RMSD, things like that. Also lets you delete files. This is one of the interfaces in Protein 3D. But if you notice the date on this model, 2/20, so 6 days before the release of this 6VSB structure, we were able to produce a model of one of the monomers of the spike protein. And the reason this is significant, and this is the model right here that we produced, the reason this is significant is because now we can go back, because there's all sorts of new models coming out, such as this 6LZG model. This model has the loop regions that were missing in that original structure. And so now we can go back and see how good did we do before we had any models to use. And that's what I want to show you today is how good was that structure we predicted 6 days before the 6VSB was released. And the way I'm going to do that is I'm just going to briefly overview this NovaFold output file, and then we're going to use some of the tools to investigate it. So this is the file that we produced around that time period. I think this was a variation we produced 2 days after the 18th. So the first model we produced is on 2/18, but this is around 2/20 that this model was produced. And you can see here, this is the size, this is the full spike protein, S1 and S2 domains. And then also we supplied a user supplied template. So we had picked out a SARS-CoV-1 model as a template, and we supplied it as a user template. That's something you can do with this tool set. But additionally, the threaders that are used in the NovaFold tool chain picked out a number of COVID-1 or SARS models that were important for its modeling efforts. And really, if you look at it, it only picked out 2 models out of the non-redundant database library that we were using for this test. And so that's interesting because we can go investigate these models now and see how they relate to the structure that we produced. Then if we go down to here, you can see this is the model overview. This is where we see the TM score, the estimated RMSD, and I say estimated, it's because we don't actually know what the structure was supposed to look like. So it's not a direct pairwise TM and RMSD. This is an estimated RMSD based on the templates that were found for this model. And then we also have the C-score. The C-score is a iTaser score metric, and all the extra scores here, those are all derived from the C-score. And if you guys care to see how those are derived, I'd be happy to do one-on-one webinars and look over the stats of all these figures. But what I want to show is how this model that we produced on 2/20 of 2020 compares to this 6LZG model that was released about a month ago. And so I'm going to click on open this new model, or sorry, open model in new document. And that's going to pop open this model. And so this is the model we produced. As you can see, it's got full loop regions because it used the ab initio modeling capability to create those loop regions. Now, if you look at this to the right here, you can see that there is no missing regions now. If we go back to this model, you can see that there's all sorts of missing regions that are grayed out. See right there and right there and right there. But this model now doesn't have those missing regions. We ab initio modeled those regions. And what I want to do quickly is show how easy it is to bring the 6LZG model into this document and do a quick TM align to see how close were we back 3-4 months before we even had a model of this RBD. So to do that, I'm going to go to the File menu. I'm going to go to the Add to Document. And the Add to Document lets you choose a file that you may have sitting on your desktop, or you can just add a PDB ID, and that's what I'm going to do. So I'm going to type in 6LZG, then press OK. That's going to connect to the PDB archives and download that model automatically for us. And then what I'm going to do is it kind of just center weights it in the middle. But then I'm going to go to the Analysis or the Structure tab, and I'm going to do Align Structure. And then I'm going to use the TM align. We also have the J-FATCAT method. These are slight variations. For today's demonstration, I'm going to use the TM align method. It just takes a few seconds. All right, so the models are now aligned. Now I'm going to deselect the selection here. And actually, I'm going to redo this. So I want to select, this is the way I wanted to show it to you. I didn't do it this way. I want to first select the A chain on my NovaFold prediction. And then I actually want to select the chain that I'm interested in. So I'm going to select the B chain and the A chain to compare to. And I'm not going to have the ACE2 receptor, which is also in this model. And I'm going to redo this alignment. Sorry about that. So now I've just aligned the spike protein that we predicted against the RBD only in this 6LZG model by doing the control click and then using the structured align structures menu. And so now if I deselect there and go to this details tab here, we can see that the RMSD of that comparison. So we had an RMSD of 1.498 angstroms in comparison, which is pretty low. So I believe that was really exciting for me when I found out that the model that we had produced nearly 6 days before the 6VSB structure was released had a really close angstrom value to the 6LZG model that was produced months and months later, and also it had the full loop structures that the 6VSB did not have. And so I was really excited about that when I found out about it. Now, additionally, if we look closely at this model, you can see that there's some very close tracing of the actual model, and this is the RBD we're looking at right here. And so if we look closely at it, we can see that the predicted model is very close visually to the model that was just done a few months ago. And so we had this months before. Now, additionally, this is the hardest region right here. This region was giving us the most trouble, but we did an extremely good job, I believe, in keeping that region the way it wanted. This would have been ab initio predicted, this region. And so you can see the gray would be the crystalline structure. The green is our structure. Those are very close in my opinion. They're very close. And so I was really excited when I saw this. And this is one way to investigate of how a predicted model compares. I know this is sort of looking back because we have this timeline of what we did and what we were working on. And but I just wanted to show that this model that was developed quite early in this COVID-19 situation looks very close to the crystalline structure. And something I was really excited about and I wanted to share with you guys today. But anyway, so another thing I want to show is that now that we know we have a good model, or kind of looking back, we didn't know we had a good model, but we have a good model. We didn't realize that until months later. But I wanted to show how we can improve when a cryo-EM structure comes out, such as this. And it's got these missing regions like this one does. I want to show how we can improve that model by bringing in a NovaFold alignment and reconstituting those missing regions. So as I showed here, is I have this model that of the spike protein. And actually, I'm going to use a slightly different model that was produced a few months ago because this model is the model I'm going to have to use for this demonstration. The early model here had a small energy minimization error that we had to correct. I'm not going to get into that, but this model here will be the one I'm going to use for this reconstituting of the 6VSB model. And so to do that, I'm going to go to open a new document first, and then I'm going to choose the A chain, the monomer. I'm going to go to File menu, And then I'm going to go to export data, export all selections. Now this will just export the A chain, and it'll actually export whatever I have selected, which is 1 through 1273, the whole thing, the whole spike protein. I'm going to save that out on my desktop, or actually in my folder here. And so now I have the monomer of the predicted model. And now I'm going to go back to my 6VSB structure, and we're going to do similar to what we did before, but actually I want to color these all back to gray. So I'm going to color everything back to gray using that style pane here. And then what I want to do is I'm going to go to the File menu, and I'm going to go back to that Add to Document. And then I'm going to load in that model, the one we had predicted, the monomer. Okay, that should just take a second to load in. Okay, there we go. All right, so I loaded in that monomer, and actually before I get too far into this, I'm going to close a few of these windows, because sometimes it can slow things down for me while I'm doing screen sharing. So just let me do that quickly. Okay, so I loaded in this A chain that we predicted, and what I want to do is I want to control click it with the A chain of the cryo-EM structure that's missing these regions. And then I'm going to go to the structure, and I'm going to go to align structure, TM align, and do the TM align on that model. So what it's going to do is align those structures together. And then I want to show you how the surface topology changed. So it just put it into that region, but as you can see, that model that we had initially, I'm going to hide some of this so we can see it a little better. There we go. So it was missing quite a bit of regions. You see that? And the region that I'm mostly concerned about is this one right here. So it was missing quite a bit of structure there. Do you see that? If I turn that on and off. And then also, if I just take this model and I apply a surface to it, and then I also take this other model and I apply a surface to it, and then I switch back and forth between them. I'm going to color this a different color so they're a little bit different colored. So now if I switch back and forth, you can see that the structures are quite different. So that's the cryo-EM structure, and then that's the structure with the modeling, with the enhanced modeling that NovaFold was able to do. So I think that's really important, and this is one of the things I wanted to really highlight today, is that we can take these cryo-EM structures and improve them with the missing regions, and these missing regions are flexible regions that are critical for what we're trying to help figure out right now. And I just wanted to point that out. Now, additionally, there's a couple other things I want to show here. So I want to show quickly how we can use the biophysical tracks to investigate other kind of things that are going on. So I'm going to hide the cryo-EM structure, and I'm just going to work with the NovaFold model now. So I have it right here. It's colored in gray. This is the RBD region. Now we can use these analysis tracks to the right here to show cleavage sites. Now these cleavage sites are really important for the function of this spike protein, and specifically the cleavage sites that I'm interested in looking at are the trypsin cleavage sites. And so I'm going to go down, and I'm actually going to just show the trypsin cleavage sites here, right here. So I just showed those only. And then we can go down to the bottom and look at those in this analysis tab. And I'm going to zoom in a little bit. And the region I want to look at is the RBD region, which is, I think it's 437 to 508. So this region right here. This is the receptor binding domain. So this is a part of the protein that has to bind to the ACE2 for attachment. And so I'm going to color that. Oh, it's over here. Sorry about that. I was looking at the wrong region. But anyway, so if we look at that region and then we compare it to this right here, this region that we're looking at with the trypsin, the trypsin cleavage sites. So we see that there's a whole cluster of cleavage sites right here, and this is really, there's some important cleavage sites that we have to understand in these regions. I believe it's actually at the 685 junction right here. So right here is where the S1, S2 cleavage site is on this, and this is what's required for the fusion protein to be exposed. And you can see that there's these 3 cleavage sites really packed together. Now this is a region that's interesting because I believe there's some evidence that that region is diverged from some of the earlier strains. Now also, I want to jump back to show this region in comparison to some of these other SARS-1 proteins. So we have this send template alignment to the Megaline Pro, and if we click that, it'll actually make a multiple sequence alignment out of all the different templates that were used to create the model. So here's the sequence that we inputted. Here's a user-defined template that we had supplied, and then here's all the choices of the threaders that were picked. And as you can see, there's missing regions and there's also insertions, and it's a little confusing because it's hard to determine if it's a missing region or if it's an insertion. But the region I was just showing you at 685, this is the region right here. And as you can see, there's quite a large insertion that occurred in this region. And this is interesting because we just confirmed that there's 3 trypsin sites at that region. And so what I want to point out is that if we're finding variations in this region, in that 685 region, if I jump back to that, that's going to affect those trypsin sites, which could affect all sorts of different things that are occurring. And so I want to quickly just paint this region, the 685 region, orange, and it's right there. And so that region is what gets cleaved off. That whole piece gets cut out using the trypsin protease. And what I want to show is that we can model variants in this region. So if your NGS data is showing that you have detected a variant in this region, and what I'm interested in is, well, how does that affect this trypsin cut site? Because if it does, then we could have a change in the way the virus is behaving. And so one of the things we can do is we can model those variants that from your ABI files or your NGS data, and we can create a variant at that position. And I'm not really sure what variant to put this to. I've looked at some articles, but if you find variants in specific regions, we can then model and see how that changes the rotamers. Also, we can see how that changes these trypsin cut sites, which I think is really going to be interesting for us. And so here I'll quickly add a trypsin and a proline, and which will most likely disrupt this region. Let's see here. But what this will do is give us a D-fire energy potential on how the energetics change in that region after applying that mutation. It can take a few minutes sometimes to do this type of modeling. It really depends on how big of a region you've selected. But when it's done, it'll give you this D-fire energy potential and a prediction if it's a stabilizing or destabilizing change that you've made at that region. Now, that's one of the things I wanted to show you because it really relates to how you're using the Sanger data and this NGS data and how we can transfer that knowledge into these PDB structures. Now, one last thing I think I still have time for is I want to add in to this document, to this model, I want to add in that one model that we just had open here, because I want to show you how we can visualize the ACE2 receptors binding location to the spike protein. And so to do that, I'm going to load up that LZG model that we had. Let's go back to it. So I'm just going to drag it in since I already have it saved on my desktop. So I'm going to drag it into the menu up here. And also what I want to do is I want to take that spike protein and I'm going to add a quick surface to it. And then I'm going to color it like I had been before, gray, to make it a little neutral color. Now, a really powerful tool that's built into this tool set, and this might be the last thing I can show you because of our time today, but what I can do is I can take that ACE2 receptor that's bound. Now, this structure was released a few months ago, so we didn't have this early on in the pandemic. But what we can do is take that ACE2 receptor that's bound and go to this select neighbors function. That's going to select, although you can't see it, that's going to select residues that are within 4 angstroms of that ACE2 receptor. And if we go to the edit menu and go to preferences, we can actually control that distance. It's set to 4 by default, but you could set it to 5 to 3. This is a way we can select a cutoff range of angstroms to select other residues that are in neighborhooding regions. And so these are all residues that would be part of the binding domain. And then what I can do is I can color that domain. So I'll color it red, and then I can hide that ACE2 receptor. And then actually we can start to visualize what this looks like visually. And that's interesting to me. Now also, we can also paint in these antigenicity tracks here. So I'm going to paint in these Jameson-Wolf antigenicity tracks by selecting them, and then I'm going to paint them the same color, this magenta color, so we can compare where these regions are. And so I've selected them already, and then I'll choose that magenta color. And so that shows us where some of the antigenicity is in that region. Now, additionally, there's other things that we can look at. We do have these T-cell epitope predictions. Also, the MHC2 complexes. I've read some articles that are looking and identifying like maybe 10 MHC2 complexes critical for vaccine development. So this also lets us use these biophysical methods to further paint and refine and visualize these models. And so these are the kind of the main tools I wanted to show you guys today. I think I'm running out of time. I think I have 2 minutes left. So I think we're gonna have to break for questions soon. But like I said, once we figured out that NovaFold was so useful for all these different capabilities, we started releasing 90-day work-at-home licenses, and all of those include a NovaFold prediction. And so if you guys need access to NovaFold or a 90-day work-at-home license, just get a hold of us and we'll deploy that to you. You'll have access to NovaFold, and additionally, you'll have training time with me if you'd like. You can email me within that 90-day time period. We'll give you as much training as you need to figure out if these tools can help you or if we can be of assistance with your projects. But thank you guys for coming today, and it was nice for me to be able to show you everything.
And don't leave, you guys. We've got some great Q&A coming up. I think we've got about 8 or so questions. I'm just going to go ahead and go through those for Thomas. So how about, okay, let's see. We'll start here. What if the protein structure is not defined in the PDB? How can you use protein 3D if there's no structure?
So if there's no structure, what we're going to have to use is we're going to have to really utilize that hybrid template ab initio modeling approach. So the benefit of the tool set, and we would go to this modeling tab, we go to protein structure prediction, new NovaFold prediction, and then it automatically entered a bunch of stuff here. But then I would be able to enter my sequence such as that in FASTA format, and it will find templates that are similar. And now the way this tool works is that the threaders look for not just sequence identity, but they look for sequence similarity using the Blossom 62 evolutionary conservation matrix. And it uses secondary structure prediction to match up similar fold types. So the tool is designed to be able to provide a structure prediction when there is no template. And that's sort of the reason this tool is powerful as it is.
Okay, thank you. The next question that we have is, how can you view the PDB file header information? I think that must be a question about the details panel.
The PDB file information, here we go. So if I click there, and if I go to the details panel, this is the information that we have, or sorry, that's not correct. Hold on.
I think it depends on what your selection is
So
too.
honestly, for that question, what I would do is I would just look at the file itself. That's what I would do. It is the most straightforward. These are just text files. So if you want to look at the PDB file, I would just open it in a text editor, and you can see that header. I mean, I'm sorry.
Yeah, I do believe that you can see that information in the details panel, but it depends on what your selection is. So right now you just have some different regions selected. If you double clicked and just expose, like had the entire structure selected, that'll show different things in the details panel. So there's kind of a subset of information that from the PDB file that will be shown here, depending on what your selection is.
All right, thanks a lot.
Okay, and another question is, has NovaDoc been used to predict protein dimer interfaces or interactions?
So we have used it for that purpose, and when we do that, we like to specify a targeted region. So that's one of the things you can do in NovaDoc, is you can specify if this is the protein docking tools interface right here. So we got a new NovaDoc prediction. But if we were to use that, we could specify receptor contacts that are known to be in the binding interface, and then it will focus the binding on those regions and find an energy minimized binding state of the two proteins. So
okay, let's see here. We've got more questions coming in. So for homology modeling, will NovaFold identify the best possible crystal structure available? So if there are multiple crystal structures that kind of apply, will it show you which one is the best?
Yeah, so in those spike proteins that I had predicted, we wanted it to be as fast as possible, so we turned off some of that functionality. But if I go into another NovaFold model, I can overview that question. Now, specifically, if we go look at the threader choices, those are the best models. So as I showed in this other NovaFold model, the best model is the choice of each threader, and each threader has a slightly different taste. But so the best model for muster was this 3-4A model. The best model, or for this PPAS, or the second best model, was this 4GER structure. But additionally, when you have the coach and cofactor methods turned on, you get more information, which we didn't have for these other models, but we get a whole listing of PDBs and their TM scores and confidence scores, coverage and identity, on if these are the top 10 models related to the model we inputted. And now those are models that were found with reverse PDB search lookups. And so that's an interesting component of the coach and cofactor methods that are part of the NovaFold toolset. Additionally, since I'm showing you this coach and cofactor method also produces small molecule binding ligand information, hotspots that small molecules were found to bind to other similar templates, such as this 3DBK. This is a reverse PDB lookup, so it finds folds of similar shape, and then it will graft in the ligand to the location specified here.
All right. Well, I'm going to just ask one more question, and then I wanted to let everybody know I'm going to be showing some training resources right after that. And any questions that we didn't get to, we're going to be making an FAQ for our website, and we will send you, everybody who's attending today, we will send a link so that everybody can see all the questions and all of the answers. So your last question is going to be, could you predict the binding residues and the strength of binding between these two? That must have happened at a particular spot in your, so I don't know. Can you predict the binding residues and the strength of binding? Okay.
And so using NovaDoc, NovaDoc provides that type of capability. So NovaDoc it can do a targeted interface search or a blind search around a protein-protein interaction, and then it'll give you a D-fire energy potential on the strength of the interaction. D-fire energy potential is a statistical energy potential based on known residue atomic interactions, and then it'll provide that information and in a probably 10 model, a listing of 10 different models and a ranking of energies that could represent different poses and different energy states of those two binding partners.
Okay, thanks a lot, Thomas. So I'm going to go ahead and take over from here. I'm going to change it so that I will be able to share my screen. Let's see here. There we go. Show my screen. Okay.
All right. Thanks, Sharon. And thank you, everybody. Hope you guys have a great rest of your day.
And so what I wanted to show you guys here, there, I guess you guys can see my screen now. Yay. I want to show you a few of the resources so that you can try out some of these things that Thomas was talking about. The first thing I wanted to show is the workflows pages. So if you click on workflows, go to DNASTAR.com, click on workflows, and you'll see there's this big list of protein analysis workflows. I'm just going to choose antibody modeling because it's at the top. So we can see this for an example. So just like all of our workflow pages, this starts out with a description of the workflow and a short video so you can see how it works. It breaks down how the workflow operates in 4 steps. We break down every workflow that we have into 4 steps. And then further down the page are resources, tutorials, FAQs, and there's also a link to the user guide here. So if we go down here, like the resources for this one, you can see some posters, watch a webinar, read some blog posts about this antibody modeling workflow. Here are some short video tutorials about different aspects of the workflow. We have FAQs, which we're going to be adding to based on some of your questions today. So thank you very much for the questions. And the other thing that I wanted to show you is in the user guide, which you can access here, or you can access through the training menu up here. Actually, let's just go to the training menu so that I can show you that. If you go to the training page, there's Protein 3D and the Nova apps. Click on there, and all training resources are in one big document. So this has every single thing that you could ever want to know about Protein 3D. You can see the huge table of contents here. So there's a lot to learn about Protein 3D, and all of the Nova apps are in here too. You can download this help document as a PDF if you want to. But the important thing I wanted to show you is that we have written tutorials with tutorial data for all of our applications. So if you click on that, you'll see here are all of the Protein 3D tutorials. So I don't know, let's choose study surface exposed cysteines. So anyhow, so here are the steps for doing that. And all of them use either a PDB file or a file that you can download from our website. And I think that was all I was going to show you. So let me just go to our last PowerPoint slide. So this has resources that will hopefully be of help. So again, here's our website, DNASTAR.com. Here are the email addresses for the 3 of us that spoke today. And most of the questions, if you have questions after the webinar, should be directed towards Thomas. We are going to take all of the questions that were asked before and during the webinar and we're going to address those in a giant FAQ. We just did those for our last 2 webinars, Seq Builder Pro and Megaline Pro, and you can see those on our blog on the website. We're going to do one now for Protein 3D and the Nova apps, so you can see everybody's questions and answers. It usually takes us about a week to get that up, and we will send you an email. Since you were an attendee, we'll send you an email linking you to that as soon as we get that up. That's all I have, and I guess it's the end of the webinar. Thanks for coming, and we hope to see you at a future webinar.
Thank you, everyone.
Bye, guys.
Have a great day.